Hi, everybody. Welcome to our presentation on fertility, pregnancy, and childbirth, the vaginal microbiome. I'm Hannah. I'm gonna start us off with our first paper. So the paper that I did, you can go to that next slide. Um, the paper that I did was Temporal Dynamics of the Human Vaginal Microbiota. This paper was written in 2012, which is the start of our journey today. So the first author for this paper is, and if I butcher this, so sorry, is Powell Jaher. He currently works as a research associate and faculty at UMD School of Medicine in their Institute for Genomic Sciences. And his primary focus is on immunology and microbiology. He actually has a really interesting background, I thought. He like completely started in mathematics from the degree that he got in Poland, and then got his PhD in mathematics from Stony Brook in New York. And then he did research on pure mathematics at a bunch of different universities for like several years. And it wasn't until 1999 that he moved into bioinformatics through a postdoc at Johns Hopkins. Um, and then previous to his time at UMD, he worked as a bioinformatics scientist at the Institute of Genomic Research in Rockville, Maryland. And so he's been working at UMD, which is where he did this paper that we're going to be looking at since 2007. So we can go to the next slide. And so to go over where we're starting today, previous findings and knowledge gaps. So previous findings that we have before this paper came out was that the vaginal microbiota is critical for reproductive health, and again, never really worked with genes before, so I'm totally gonna butcher these names, but lactobacillus species playing a key role in preventing infections. Additionally, women's vaginal microbiomes can be categorized into five categories that we're going to talk about through these papers today, that are known as community state types, so CSTs, with some dominated by Lactobacillus crispatus, L. gasseri, or L. inners, while others are more diverse. At this point, most of these studies that were done were cross-sectional, meaning they took samples at a single point in time, um, which do not capture like the temporal changes in microbial composition, as we'll see that's part of our knowledge gaps. And bacterial vaginosis, also known as BV, is linked to an imbalance in vaginal microbiota, but the cause of stability and transitions between microbiome states remained unclear prior to this research. And so our knowledge gaps are the lack of longitudinal data, unknown stability and transitions of the vaginal microbiome, and limited understanding of microbiome resilience. So, and we didn't really know at this point whether shifts correlated with menstruation, sexual activity, or other factors. And so we're going to see how this paper helps us understand those things right now. So, on the next slide, we'll see our methods. So, the methods for this study, basically, in terms of population, it took 32 healthy reproductive age women. And for the longitudinal part of the study, it was 16 weeks long, and they had twice weekly vaginal swab collections. Um, they did microbiome sequencing using 16S rRNA gene sequencing to classify bacterial species, and then they did metabolomics, maybe is how you say that, analysis using NMR spectroscopy to assess metabolite production. And then the tracking factors influencing stability that they had their participants report were menstruation, sexual activity, douching, and lubricant use. And then three different statistical and computational analyses were used in this paper. The first one being Jensen-Shannon Divergence Index, which measured microbial stability. The second was hierarchical clustering, which classified the vaginal microbiome states. And then the last one was principal component analysis, or PCA, which was used to analyze metabolite patterns. So on the next slide, we're going to look at our first figure, which is going to be showing us the vaginal microbiome community state type, so the CSTs and transitions. And this figure kind of shows us how the different microbiomes shift over time. And so there's five major vaginal bacterial community types, and they were confirmed through this study. The first was CST1, which is Lactobacillus crispatus dominant, which is the most stable bacterial community. We have CST2, which is Lactobacillus gasseri dominant, which is for rare transitions. CST3, Lactobacillus inners dominant, which is moderate stability. And then we have CST4A and CST4B. CST4A is mixed composition, some lactobacillus, but also anaerobes. And then CST4B is highly diverse, includes Gardnerella, Prevotella, and Atopobium. And this is the one that's linked to BV. And so we also saw transition states that were pretty common. And so CST1 mostly transitions to CST3 or CST4A. CST4B often transitions to CST3, but rarely to CST1. And then CST1 is the most stable and rarely transitions. And so this figure basically shows us which CSTs are stable versus which ones frequently transition. And it also is providing insight into why some women may be more susceptible to infections, pregnancy complications, or recurrent bacterial vaginosis, aka BV. So that's why this information is super useful. And then if we pop down to the next slide on figure 4, this one is showing us the menstrual cycle influence on microbiota stability. And so this figure is demonstrating that microbiota are least stable during menstruation. And this figure was produced using the Jensen-Shannon divergence index that we just talked about. And so menstruation, basically, this proves that menstruation was the biggest disruptor of vaginal microbiota stability. And our two key findings from here are that some communities are resilient and return to their original state, whereas others shift permanently to a new CST, particularly CST4B, which again is the one that's goes along with BV. And yeah, that's pretty much the big things we gain from here. Also, it's really important for us as all women cycle. Well, women, people with uteruses. So it's important for us to understand how the cycle affects the microbiota and how this can help timing-based interventions that we would look to in the future to treat any sort of things that we relate to the vaginal microbiota. And then in the next slide, we can look at figure 5, which this might be hard for you to see. So, because it is such a big graph, so you might want to reference this on your own device and have like the graph pulled up. But this figure highlights how different microbiomes maintain metabolic function. So it's all about functional stability despite microbial changes. So these graphs show that even when the microbial composition changed, the metabolome, which is basically a fancy word for the functional output, remains stable. In addition, lactic acid production was preserved even when different lactobacillus species fluctuated. Also, CST4B, which is again that BV related CST, these communities had higher levels of succinate and acetate, which are linked to, again, bacterial vaginosis. And then lastly, some transitions from L. crispatus dominated to L. inners dominated states did not disrupt function, suggesting functional redundancy in the microbiome. So basically, like takeaways from this figure is that microbial composition changes don't always necessarily mean dysfunction. So as long as the microbiome continues producing lactic acid, it can still protect infections, which is super huge. And also, CST4B produces more succinate acetate, which is linked to bacterial vaginosis and inflammation. So here we get that direct tie between that CST state, and BV. And so, going to the key findings for this study is that the vaginal microbiome is highly dynamic with menstruation as a major driver of change. And so, menstruation disrupts microbiome stability more than sexual activity or hygiene practices, which is super interesting. Instability during menstruation can actually lead to transitions toward CST4B, which can increase infection risk. So, that's a super huge finding. Um, Next is that stable microbiomes tend to be dominated by Lactobacillus crispatus, which while diverse microbiomes fluctuate more. And so CST1 and 2, which are the lactobacillus dominant states, are stable and protective, whereas CST3 and 4B fluctuate more frequently. And so frequent transitions between CSTs can increase susceptibility to BV, infections, and reproductive complications. So that's why it's super important for us to be able to find this. And if you can't kind of already tell, I'm going to speak on this in a second, but everyone's experience and everyone's transitions are very unique and individual, which is a huge part of why this study shows us we really need to be individualizing vaginal health. But more on that soon. Um, a third one is sexual activity has a small effect on stability, like I just said, but it does not necessarily cause dysbiosis. So, unlike menstruation, sexual activity leads to only mild microbial shifts, and most transitions are not long lasting. So, you're not going to see an increase in infection risk with things like sexual activity. Next, microbial function remains stable despite bacterial shifts, suggesting functional resilience. So even when composition changes, vaginal health can be maintained if lactic acid production remains stable. And in CST4B, um, along with this is just all about more about how CST4B is linked to BV. Then high microbial diversity is not always a sign of disease, challenging traditional diagnostic methods for BV. And this is a huge reason why this study was super important because cross-sectional studies previously done before this one missed this complexity. And this longitudinal study really allowed us to track um, all things that are necessary to truly understand vaginal health and really just zoom out and look at the bigger picture. Um, and so that's super great. Because the traditional BV diagnostics like really assumed that high diversity was unhealthy, but some women with diverse microbiomes remain asymptomatic. So again, another indicator that vaginal health is very individual. And then all of these just the implications for fertility, pregnancy, and women's health. So microbiome instability is linked to preterm birth and miscarriage risk for some women. And so tracking microbiome shifts over time could improve reproductive health interventions. And so then the last slide for this study is on critiques and future investigations. So some critiques of this study is its strengths was, which I've been saying a bit so far, is that the first long term, it was a first long term study tracking twice per week, which is super important for that bigger picture, like I was saying. Um, it used a really comprehensive approach because it integrated genomics, metabolomics, and behavioral data. So that was really interesting. And then lastly is its use of it really introduced the Jensen-Shannon Divergence Index as a new way to measure microbiome stability. So that was really cool. And then it did have some weaknesses. So obviously only 32 women is a very small sample size. Um, it also did not include pregnant women, which is important for understanding microbiome changes in pregnancy. It did not control for diet probiotics and antibiotic use, which could definitely influence microbiome shifts. So that's definitely something to look into in the future. And then it only assessed um, bacterial composition and did not analyze host immune responses, which could also impact microbiome stability. And so lastly, for future investigations, I would love to personally see again more research on microbiome changes specifically in people who are pregnant. Um, Additionally, anything linking microbiome changes to clinical outcomes. So, how do these microbial shifts influence fertility, miscarriage, or preterm birth? Intervention strategies. So, can probiotics or microbiome targeted therapies help stabilize vaginal communities? That would be interesting. Um, obviously, more large and diverse populations. This study very much showed that Vaginal health is very individual, so I'm curious to see like how this can change depending on the population we're looking at. And then lastly, like I've been saying, personalize how like more investigations on how this research impacts personalized vaginal healthcare. Not all microbiomes need intervention, so treatments should be tailored and individualized to the person. And yeah, that's all for my study.
Okay, so the next paper is ovarian cycling and reproductive state shaping, or shape the vaginal microbiota in wild baboons. So the primary investigator was Elizabeth A. Archie. She's a professor at the University of Notre Dame, and the director of Baboon Research Project in Kenya. She got her PhD at Duke University, in biology, and then most recently she was awarded external funding, to develop insertable cardiac monitors to assess social and environmental effects on the autonomic stress response in a non-human primate model of aging. And then the previous findings to this study is the composition of the human vaginal microbiome varies considerably between individuals and over time. Human va vaginal microbiota is typically dominated by members of the Lactobacillus genus, which makes up about 70% of the resident bacteria, more specifically lactobacilli. The loss of lactobacilli dominance results in increased pH and bacterial vaginosis, which is again associated with infertility, preterm birth, maternal infections, and increased risk of STDs. Um, on more previous findings is that factors influencing human vaginal microbiome um, include estrogen levels, reproductive state, menstrual cycle, sexual contact, and exposure to maternal bacteria. Although in non-primates, they have fewer lactobacilli. Less than 2% of their resident bacteria is lactobacilli, and they have much higher diversity in their vaginal microbiome. So, what this study aims to answer is how does the reproductive state, vaginal pH, and the ovarian cycling affect vaginal microbiota in wild baboons? Does sexual behavior influence vaginal microbiota? And is there vertical transmission of vaginal microbiota in baboons? How does vaginal microbiota in baboons compare to those observed in humans? And what shapes the inter-individual differences in vaginal microbiome of baboons? Okay, so the methods of this study. The study subjects were 48 adult female wild baboons in Kenya, and some of the baboons were sampled twice. So they took the sexually mature female wild baboons and they performed 52 vaginal swabs collected between 2007 and 2010. And then they performed DNA extraction and sequencing and then ran statistical analysis like the alpha diversity, which is a multivariate linear regression model to test possible predictors, and then beta diversity, which identifies predictors of the similarity. And then they tested the horizontal and vertical transmission, as well as the taxa identification and vaginal pH. So in the first figure, this is basically just showing how the progression of reproductive states and the ovarian cycle phases in female baboons were identified. So there's four ovarian cycle phases, such as swelling, perioovulation, detergences, and anestrus. And then the reproductive states were pregnancy and postpartum. By looking at the perineal skin or the pericallosal skin, um, you can determine what phase or states the baboons are in. So in figure two, of the 29 phyla, 11 phyla were found in all samples of the swabs, and then 43 genera were also found in all samples, including multiple genera that have been linked to BV in humans. The median relative abundance of lactobacillus was only 0.00063 in baboons compared to the reported 96% in humans. Okay. And then this is just showing the Shannon Diversity Index in the OTU richness performed. Um, Basically, the reproductive states uh, show that cycling females had the lowest vaginal microbiota diversity, and postpartum female had the highest vaginal microbiota diversity. The ovarian cycling, the anestrus females had the highest diversity, and then social group size, the larger social groups resulted in lower microbial diversity, potentially due to more sexual activity. And then for rainfall, the months with the most rainfall correlated with lower microbial richness, which is just an implication of environmental factors. But again, correlation doesn't equal causation. And then the key findings are, according to the study, there is no strong evidence of vertical transmission of vaginal microbiota from mother to offspring in baboons. And unlike human vaginal microbiota, female baboon microbiome is not dominated by lactobacilli. Reproductive state and ovarian cycling plays an important role in female baboon vaginal microbiota composition. Sharing sexual partners contributes to vaginal microbiota similarities between baboons, and multiple genera linked to BV were found in all samples of female baboon. And then some critiques is that this was a limited sample size of just 48 baboons, and some were even swabbed twice. The subjects also came from the same ecosystem, and there could be selection bias as to which baboon is best for this study. And then the correlation does not always equal causation, like the rainfall study. And then pH measurement was only measured within the span of 1 year. Future investigations could be more short-term testing, tracking microbiome to successful conception, investigating the role hormones may play in vaginal microbiome, and then investigating microbiomes and susceptibility to vaginal infections.
All right, the next paper was the maternal vaginal microbiome partially mediates the effects of prenatal stress on offspring gut and hypothalamus. And Tracy Bale got her PhD from the University of Washington, Department of Pharmacology, and then she went on to do her postdoc at the Salt Institute. Then she worked at UPenn as professor of neuroscience for 15 years before she was recruited to the University of Maryland to develop the Center for Epigenetics Research in Child Health and Brain Development. And now she's at the Department of Psychiatry at CU School of Medicine, and her research focused on stress dysregulation and neurodevelopment and neuropsychiatric diseases. The previous findings of this study, they knew that maternal stress during pregnancy is a risk factor for neurodevelopmental and GI disorders, and that also maternal stress alters the microbiota composition that colonizes the neonatal gut. And then they also knew that the pioneer community of maternal microbiota in babies' GI tracts at birth has a lasting impact on their gut and brain. So they wanted to see if the maternal vaginal microbiome mediates the effects of prenatal stress on offspring and how postnatal development would be affected by the transfer of stress-altered microbiota composition at birth. So figure 1A is basically how they did this experiment. They did C-section delivery and neonate oral garbage to manipulate the colonizing microbiota. And so they did this to see if maternal vaginal microbiome mediates the effects of prenatal stress. So CC is when it's a controlled dam transplanted to control offsprings. CS is when the microbiome from the controlled dams is transplanted into stress-exposed offsprings. SC is stress-exposed dams to control offsprings, and then SS is the microbiome from stress-exposed dams to stress-exposed offsprings. They also cross-foster the offsprings in surrogate dams, so they're with both sexes and all treatment groups in each litter to control for some of the postnatal environment and housing conditions that may be confounding variables. And then figure two showed that figure 2A and B is looking at body weight, and that was measured from P14 to P70. And you can see that the growth curve for the the SS male offspring is lower than the control male, and that replicates findings from previous studies. But the interesting thing to look at is that there was reduced body weight of the C to S male compared to the C to C, but no difference from the C to S and the S to S. And there was also no difference observed in female offspring groups for either body weight or cortisone levels, which was interesting. And then figure 2D is stress-induced cortisol levels, and they again found the same things that S to C males had increased levels compared to C to C males, but not significantly different from S to S males. And so both of these suggest that there's an intermediate phenotype and a possible mediating effect of the maternal vaginal microbiome. The key findings of this paper was that the maternal vaginal microbiota modulates effects of prenatal stress on neonatal gut microbiota composition, body weight, and stress response. They also found that prenatal stress changes the fetal intestinal niche in male offspring, and that may determine which microbiota can reside in the offspring's gut postnatally. They also looked at how chronic stress in adulthood unmasked these effects of the early prenatal stress on the gut. They found prenatal stress is associated with altered gene expression in the paraventricular nucleus of the hypothalamus of males exposed to adulthood stress. Some critiques for the paper is that stress during pregnancy also affects other things other than the microbiota, like the mother's hormones, for example, cortisol, the immune response, and their diet. And those could all be confounding variables. And so there's not really a good way to tell that this is solely based on the microbiome. And they also only looked at the vaginal microbiome and ignored the gut microbiome, which may have to do with this. And for further research, they need to replicate this in human studies and see if there's clinical correlations. I thought it'd be interesting to study the vaginal microbiota of mothers with children now who have neurodevelopmental disorders and look for correlation there. And then it would also be interesting to test possible interventions like a probiotic supplement in different stages of development in both the mother and the offspring and see if those have any effects.
Hi everyone. So today I'm going to be doing the study on rectal and vaginal eradication of Streptococcus algactae GBS, which is found in pregnant women by using Lactobacillus salvarius sect 9145. So Lactobacillus salvarius sect 9145 can possibly help reduce group B streptococcus, which is GBC, colonization in pregnant women. So this study published in Nutrients by Martin in 2019 looks at an alternative to the current preventative strategy of interpartum antibiotics. Antibiotics are effective, but they carry the risk of microbial resistance and distribution of infants' microbiome. However, the study shows that probiotics may be safer and a more natural way to control GBS colonization. So in this presentation, you will see why. So Dr. Virginia Martin is the author of the study. She is a researcher in microbiology and probiotics with a special interest in maternal and neonatal health. She's also the associate dean of the Department of Nutrition and Food Science in Complutense at the Complutense University of Madrid. She has been involved in some studies concerning the prevention of infections by some by probiotics. Dr. Martin has also previously studied the development of probiotic-based interventions for bacterial infections with special interest in pregnancy. She has also published her research in high-impact journals based on microbiology and food science, and has worked with leading authorities in the field of microbiology and food science. She has also helped add to the growing body of evidence that probiotics can actually be used in the place of antibiotics to fight colonization for group B streptococcus, which is GBS, sorry. So to go into some previous findings and knowledge gaps, literature review invoked earlier learning that the rate of GBS colonization is actually high and often results in neonatal infections. Antibiotics are currently used in preventative measures, but they are known to disrupt microbial development in infants and exacerbate resistance. So, in fact, there are some promising results associated with certain probiotic strains. However, they're not definitively proven. So this study contributes to the available knowledge by assessing L. salvarius sect 9145 for the control of GBS, but there are still concerns as to the pleiotropy of long and short-term effects of such agents, which include continuously dosage and working with or the working of probiotics. However, studies on the effect of probiotics within preterm babies, neonatal health, and diet factors are sought out to be conducted to enhance these strategies. So going into the methodology of this study, the paper was a prospective clinical trial. So it discussed the trial, including 57 pregnant women that were randomly assigned to the probiotic group, GBS positive control group, and GBS negative control group. Study participants belonged to the probiotic group received L. salvarius sect 9145 on a daily basis through weeks 26 to 38. So screening for GBS was carried out by the means of vaginal and rectal swabs of at the 28th, 32nd, and 38th week. Identification analysis showed bacteriological changes, and parametric tests quantified the difference over time. The research also has an immense potential to be used for large-scale research for the validity of additional concepts to be tested and affirmed. Going into the next slide, so this figure one, the first, so this picture depicts the L. salvarius as an anti-adhesive that attaches to GBS directly and inhibits its ability to settle to the vaginal tract, which makes it relevant to the microbiome functioning by providing the necessary substrate for bacterial action. So figure 3 indicates that GBS concentrations are negatively associated with probiotics and demonstrate the progressive reduction from 26 to 38 weeks. The lack of change in the control groups that did not receive L. salvarius demonstrates the impact of the probiotic. So as we can see in this table right here. So going on to figure 4. So the safety of L. salvarius sect 9145 in pregnant women has been demonstrated in this figure. As you can see, no side effects were observed amongst the participants, meaning the taken probiotic did not have any negative effects. There were no significant changes in maternal weight, blood chemistry, and health, which also shows the probiotic effects. The probiotic is also very safe in view of the findings of animal testing, which further encourages its application during pregnancy. These findings support that L. salvarius as a probiotic could be used. However, further large-scale research is required to ensure the safety of this compound among various ethnic groups to establish any harm that may be caused to other groups. So biological mechanisms, just to get a brief overview, L. salvarius decreases vaginal pH by producing lactic acid and inhibits GBS growth. It also exudes antimicrobial compounds such as hydrogen peroxide, which also kills them. So GBS also competes for adhesion sites in the vaginal and intestinal mucosa and improves the overall immune response that the body has and helps balance the microbiome overall and creates unfavorable conditions. So key findings, overall, the outcomes of the present findings show that the use of L. salvarius diminishes the levels of GBS colonization in pregnant women. And finally, at the end of the trial, a group of women who had taken probiotics tested negative for GBS by 72%, while vaginal GBS was negative by 68%. In this study, the control groups were not able to demonstrate any decrease in GBS colonization. The statistical test also showed that bacterial count decreased over time, which proved that probiotics do work. Also, there were no side effects reported, as discussed earlier, which indicates that L. salvarius is safe for consumption. These observations indicate that probiotics may be useful in promoting GBS colonization, whereas the use of antibiotics has shown to be counterproductive in preventing GBS transmission during pregnancy. So critiques, despite the fact that the findings of the present study are encouraging, it's worthwhile to examine some of its limitations. So the study has a small sample size of 57, which means that other similar studies are necessary for confirmation, particularly studies with a larger population size. Also, long-term efficiency of L. salvarius is established for both women during pregnancy and newborn remain still unknown. Future studies should aim at establishing whether or not there is any disputes later down the road for infants as they develop. And yeah, and that is it for this paper and for our presentation. Thank you guys for listening.